Okay, next one. Are we there yet? No, not yet. But it's clear now, right? We can only have one name for each species of fungus, whether or not it's a telomorph or an anomorph. So now you have to be very careful. Bad taxonomy can kill. So pag nakakita kayo ng mushroom, ayusin nyo identification nyo. At saka pangalan, baka mamaya pagkain nyo, naku, either mag-hallucinate kayo kasi ang nakuha nyo clitocybe or psilocybe, yun yung mga magic mushroom. Or yung mga nagtatai kayo kasi masama, yung mayroong toxic na mushroom, yung mga amanitas. Yung katulad nitong aking drawing, amanita yan. So, minsan, meron kasi, usually, kaya bad taxonomy, marami siyang doppelgangers yung mga look-alikes. Na hindi naman talaga siya yung masama, pero kasi kamukha siya. Kaya you have to be very careful talaga. Okay, so navigating through the confusing changes in fungal taxonomy. Kahit kami mycologist, marami kaming nakakalito pa rin. Maraming nagko-complain sa amin. Ano ba yan? Bakit ba yan lagi na lang nag-change? So, the next slide, JP. So again, let's review. So the traditional classification was based on sexual reproduction, right? So then in 2007, next one, please. In 2007, there was a phylogenetic assessment. So now we're talking molecular. So what they did here, so this was Dr. Hibbett, a very well-known mycologist, and his cohorts. So they did a very big phylogenetic assessment on all available published data, sequence data. And the results showed that many of the species, so ito yung pre-2007, nakita mo, simple lang sila. 4 na phylum, 4 lang na phyla. Nung ginawa nila ng assessment based on sequences, marami, for example, here sa Glomeromycota, yung green dots, maraming lumabas na hindi pala sila related dun sa Glomeromycota. So they went out of the group. Yung sa chytrids naman, merong isa, yung Blastocladiomycota. So yun ang nangyari. So yung dating morphology na classification, it wasn't the true classification. Yung hindi siya natural. So ito kasi, sequences, genetics na to, natural classification. So, ito na, this is what we are going to follow. Okay? And they also saw that Microsporidia, which was a spore-forming parasite that were once considered protozoan, are actually fungi. So it was very, very surprising. But Yahoo moment talaga yon. Marami kaming Yahoo moment sa fungi. You should go into mycology. I'm telling you. It's a very, very interesting thing. Last frontier para sa amin yon. Di ba? Parang sa Star Wars. Dami-dami. Ang dami-dami kasing species na hindi pa alam eh. Kaya very exciting talaga. I'm excited. Anyway, so next slide. So that was in 2007. And then in 2018, another phylogenetic assessment was done again. And then those species that were taken out of their respective groups were assessed again. And then more data came in kasi to 2007, 2018, ang dami na talaga, as in sa GenBank, which is a database of sequences, we have now, as of probably December, 200 million sequences. Pero ang daming data yon, and it's for us to use. It's free. So that's a really good thing. So now, from one subkingdom, yung subkingdom Dicaria, ngayon naging 9 na. So this is now our current classification. So we have still the dicarya, and then those that came out of the glomeromycota, yung mga mucorales, meron na silang sariling phylum. Meron na subkingdom. So Mucoromyceta. So naging 9 na from 1. And this was done in 2018 by Su et al. Na parang European tong grupo na to. Okay. So, why were changes needed talaga? Why did we have to change? Because, you know, yung traditional classification was based mainly on morphological species concept, so yung morphology. But ngayon, because now we have demonstrated that morphology did not represent a natural grouping, then now, because of DNA sequencing, we can see that they're really divergent. For example, species A, B, and C, you know, Penicillium, by morphology belongs to the genus Penicillium. And then, nung nag-analyze kami ng sequence variation, species B, yung sa Penicillium, went out. It's found to be phylogenetically close to Thalaromyces. So, nangyari ngayon, because ngayon, doon na siya ika-classify sa Thalaromyces, kailangan na niya i-change ang name niya. So, hindi na siya Penicillium, magiging Thalaromyces na siya. So, yun yung pinakita ko kanina, yung Thalaromyces marnifii. Bakit siya na-change from Penicillium to Thalaromyces marnifii? Because of that, when they phylogenetically, when they assessed it with the sequences, nakita nila na hindi siya talaga related sa Penicillium. So, see, the genes are talking now. So, kaya karamihan na ngayon, mga gene junkies naman. Wala nang ginawa kundi mag-sequence. Hindi na tinitingnan yung fungi. That is not correct. Kasi you still have to know your fungus. Hindi pwedeng basta na lang sequence. Oh, alam ko na, A, C, B, andun na sa Thalaromyces. Pero why? Do you know? Do they look the same? And they do. Kasi yung Thalaromyces, para siyang Penicillium, but they produce this very, very matingkad na kulay. May red, may orange, may yellow. Yun ang characteristic ng Thalaromyces. Kaya makikita mo talaga ang Thalaromyces marnifii, very red siya. Meron siyang red na reverse. Ang very beautiful, pero very deadly. Very deadly, deceptive talaga. Okay, so next one is, so then why were changes needed? So the discovery of cryptic or sibling species that lack distinctive morphologic characteristics within existing phenotypic species. So here's an example. For example, yung Aspergillus fumigatus, and I'm doing this as an example kasi parang everybody probably is familiar with Aspergillus, right? So Aspergillus fumigatus, meron akong mga 50 na isolates. So out of the 50, merong 10 na katulad siya talaga. It looks the same as Aspergillus, pero hindi siya nag-grow sa 50 degrees. Pero nag-grow siya sa 10 degrees na temperature. So when they assessed this 10 strains or 10 isolates, they found that it was really a different species. So maraming ganyan. And why was it important? Because nakita din namin na ang kanyang susceptibility to azole and echinocandins ay nabawasan. It's lesser. So resistant siya. Imbis na with fumigatus, you can use azoles and echinocandins. Okay siya. Mag-work itong mga antifungals na to. Pero yung lentulose, hindi. Refractory siya. Hindi siya susceptible. So, hindi mo siya matitreat ng azoles. Okay, so now we have to describe that as a new species because it's very important that we know what that is so that people will know or the clinicians will know how to treat it, what antifungal drugs to use. Okay, and again, as I've mentioned before in the previous slides, the same fungus can occur in many different forms, so sexual and asexual. And then many have been described and named at different times without the teleomorph-anomorph connection being recognized. Pero ngayon, without the structures itself, we can connect by using the sequencing. So ang ginawa ko, kung isi-sequence mo yung Arachnomyces at saka Onychocola na species, they should have identical sequence. Kasi pareho dapat sila na species. So that's how you can connect the teleomorph and anomorph connection without actually looking at the structures. So now, kung hindi mo ma-connect yun, magiging confusing talaga kasi magkakaroon ng dalawang names. Okay? So this is now our mantra, yun. Phylogeny Outranks Taxonomy every time, anytime. Talagang kung magsasalita talaga, pag papapiliin ka, phylogeny talaga is the correct one as opposed to morphology. Kasi makikita mo sa example ko ngayon, makikita nyo na why is it like that? Why is it related to that? Doesn't look like it, right? So this is my example. So Aspergillus. So after the name changes, so they said that, okay, we will have one name, one fungus, one name. So my good friend Hubka, Beat Hubka in the Czech Republic. So he in his lab, they did a very big assessment of phylogenetic assessment of Aspergillus. And you know, Aspergillus is a very specious genus, meaning there's hundreds of species, very big, like Fusarium, very specious. So what they did was they did the assessment together with closely related genera. So for example, anamorph genera like Dichotomomyces, Polypacillum, Phylosimplex. Actually, we described the Phylosimplex as well. And then teleomorph genera, Neocytoria, Emersella, Eurotium, Petromyces. So noong ginawa nila yung assessment, nakita nila na it's monophyletic, meaning it's under one clade or one group with one ancestor. So they said, okay, they're closely related. So now, because of the one name, one fungus, one name, we will list them under Aspergillus. But tingnan nyo naman, look at the structures of these. See, it doesn't look like an Aspergillus. So when you look at this in culture and you don't know that there were some changes, you would still say, oh, I'm seeing a Polypacillum. No, it's not. It's Aspergillus now. So nowadays, people are getting to be educated with these new names. So that's our role as the lab, the identifying lab, to tell them that no, it's hindi na yan yung name ngayon. So for example, if I identify it as a Phylosimplex, for example, then I would say formerly known as, so that they would know that it was named this, but now it's currently named this. So kasi minsan, hindi naman minsan, most of the time, they will look up the name of the fungus or cases or infections para makikita nila yung mga ginagamit na antifungal drugs. So, but if they don't know which name to look up, hindi nila makikita yon. Okay. So now, because of that, we have a framework for going forward. So because mycology is very big, there are working groups. For example, there's working groups for Aspergillus, for Fusarium, for dermatophytes, yun yung mga skin fungus, yung mga tinea, yung mga anong tawag doon sa Tagalog, mga an-an, yung mga ringworm, yon merong isang grupo yon. So, because it's so big, so they can focus on those groups and then they can do the taxonomy, whatever it is that they need to correct. Okay, so there's a lot of transfer of species from one genus to another and then renaming. Diyos ko, malilito ka talaga, maluloko na ka. Pag meron kaming ina-identify, then it's my job to know whether the name is current or not. Otherwise, ako yon. Ako ang responsible doon. Okay. So I'm going to give you an example how and when to describe a new species. So next one, please, JP.